Standard biology defines cellular life through the Hayflick limit. Somatic cells possess a finite replicative capacity. Every time a cell divides, the telomeres protecting its chromosomes shorten, and once they reach a critical minimum, the cell locks into senescence. We are now seeing a framework where cells do not age in isolation. Instead, they operate within a herd rejuvenation model. In this network, specialized immune cells share genetic vitality with distant, deteriorating tissues to reset their biological age. This provides a mechanism for the effects seen in heterochronic parabiosis. When the circulatory systems of a young and an old mouse are surgically joined, the older animal undergoes rapid, multi-organ tissue healing. Traditional intracellular telomerase activity is insufficient to explain this level of organism-wide regeneration. The math of localized repair cannot account for the scale of the recovery. These findings indicate that biological aging is the result of a breakdown in a systemic genetic subsidization network. Researchers at Oxford University and the firm Sencell recently isolated the specific factor in the bloodstream responsible for this systemic repair. They identified telomere rivers, structured circulating lipid vesicles that carry physical telomeric DNA and a complex molecular payload. The adaptive immune system, specifically the CD4-positive T lymphocytes, acts as the primary manufacturer and systemic distributor of these rivers. This identifies a broader purpose for the immune system. Beyond its role in pathogen defense, it functions as the master engine for tissue maintenance across the entire organism. These youth factors are generated during an active immune response. It begins when an antigen-presenting cell docks with a T cell, forming an immunological synapse. Within the antigen-presenting cell, calcium triggers the degradation of the sheltering complex capping the chromosomes. The enzyme TZAP cleaves the exposed telomeric DNA. This DNA is packaged into a vesicle alongside Rad51, then transferred across the synapse gap directly into the recipient T cell. This links an organism's ability to mount an immune response to its capacity for systemic repair. The T cell processes this genetic material through a specialized asymmetric division, producing a subpopulation of stem-like memory cells. During this division, the T cell applies a strict proteomic filter, segregating telomeric vesicles away from its glycolytic machinery, specifically GAPDH. While removing glycolytic markers, the T cell enriches the vesicle with reprogramming factors like Notch1 and beta-catenin. This filter is a biological necessity. Delivering glycolytic enzymes to aged tissues could inadvertently trigger chronic inflammation or the development of malignant cells. This curation ensures telomere rivers function as a safe delivery system for tissue reprogramming. River formation relies on a specific metabolic state. The T cell must be actively performing mitochondrial fatty acid oxidation. The gatekeeper for this pathway is the enzyme CPT1A. Youthful T cells use CPT1A to drive river production, while aged cells stop, accumulating toxic ceramides. Fatty acid oxidation clears these lipids, generating the phospholipids required to stabilize the immunological synapse. This explains the impact of inflammaging. Chronic systemic inflammation destroys the T cell's ability to oxidize fatty acids, effectively breaking the CPT1A gatekeeper. This identifies metabolic dysfunction as a primary driver for why an aging organism stops repairing its tissues. Researchers at institutions like the USC Leonard Davis School of Gerontology have investigated how to manually override this metabolic gridlock using the fasting-mimicking diet. The protocol's depletion phase drops IGF-1, allowing the FOXO3 transcription factor to enter the nucleus, initiating cleanup of damaged white blood cells. In the refeeding phase, nutrients activate mTORC1, driving massive stem cell proliferation to repopulate the immune system. This biphasic intervention replaces exhausted cells with a naive population capable of restoring the CPT1A-driven fatty acid oxidation required for river production. Targeted metabolic stress successfully reboots the immune system's capacity to maintain the systemic river network. In murine models, the administration of purified telomere rivers or the use of metabolic reprogramming resulted in a 17-month median lifespan extension. The result exceeded standard life expectancy. The treated mice survived past the known biological ceiling for the species, with some living to nearly 5 years. Human clinical trials of the fasting-mimicking diet have demonstrated a 2.5-year reduction in biological age, measured by clinical chemistry markers predictive of mortality. The most significant finding was the persistent reversal of immune aging, specifically restoring the youthful lymphoid to myeloid ratio in participants over 40. These results indicate that biological aging is not a fixed trajectory, but a dynamic state that can be systemically reversed. This mechanism is not unique to mammals. Researchers have successfully isolated identical telomere rivers from the xylem sap of orchids. Just like mammals, plants use the exact same metabolic trigger, photosynthesis-driven beta-oxidation, to carefully package and circulate these powerful youth factors throughout their systems. Intercellular genetic subsidization is a primordial survival program that evolved to maintain the health of complex multicellular organisms. Chronological time is constant. But biological aging is a metabolically gated network, and we now possess the framework to manage it.